Good evening. Welcome everyone and thank you for coming. If you don't know me, I'm Michael Yaffe. I'm a faculty member here at the Koch Institute. I'm the director of our MIT Center for Precision Cancer Medicine, and I'm a professor of biology and biological engineering. And I moonlight as a surgical intensivist, emergency surgeon, and surgical oncologist. I'd like to thank all of you for coming. I'd like to thank the MIT Club of Boston for co-hosting this evening's program with us, and particularly Megan Jędrzejczyk for her help. Tonight's program, Lethal Weapons Against Cancer, Targeting Cell Death and Injury Pathways to Eradicate Tumors, is part of the Koch Institute's With Insight public event series. These events are an opportunity for us to share the cancer research insights that we're pursuing and the progress within sight with the public, as well as with members of our local clinical, biotech, and MIT alumni communities. Over the next hour, we'll be talking about cell death and injury pathways. I'll start by giving a general overview and background on some of the work in my own lab before we hear from Dr. Tiffany Emmons, a postdoc in my group, about one of our current projects that she's leading. Then we'll hear from my colleague, Professor Whitney Henry, who works on a specific cell death pathway to find new ways to target the most aggressive cancer cells before we open up the floor for questions. This beautiful image, which is featured in the event's materials, comes from the work of Dr. Tiffany Emmons in my lab, who you'll be hearing from shortly. To open this evening's presentation, I would like to start with a big question behind tonight's event, which is, cancer treatments kill tumor cells. Does it really matter how they die? Now, as family members, friends, and loved ones of cancer patients, or as patients ourselves, we might be inclined to say, absolutely not. A dead tumor cell is a dead tumor cell. But as physicians and cancer researchers in this community of people gathered here who are interested in the broad progress against cancer, we would argue that yes, it actually does matter. As we're learning, the cell death and injury pathways that are activated by cancer treatments can have a big impact on how effective those treatments are against certain cancers, or against certain types of cancer cells, or in how we should be combining cancer therapies for different patients, either at the same time or over the course of their treatment. Let me take a step back and give you a quick primer on cell death. This diagram is just meant to give you a sense of scale. We don't need to go into it in detail about each type of cell death, except I will tell you that when I started in this field, there was exactly one type of cell death that we knew about, and that was called necrosis. That just meant the cells were really dead. And then over time, we realized there was a second pathway called apoptosis. But now the number of cell death pathways continues to enlarge. In fact, we've uncovered so many new cell death pathways that it's changed the way we think and talk about cell death. There's been a shift from our original way of thinking about cell death, which was really categorized in terms of what the dead cells look like. Was it shriveled up like a raisin? Did it splatter debris all around? Into an entirely different way of thinking about how these types of tumor cells or normal cells die. Now our paradigm has shifted to also include molecular mechanisms in triggering insults. And again, this is just meant to give you a visual picture of our thinking. This primarily deals with how pathogens cause cell death, but it should give you a sense of how our understanding has now broadened to include both pathways downstream of stimuli and different types of immune cells that are contributing not just to the morphology, but to all of these other factors that determine the precise way that tumor cells die. Understanding the cell death mechanisms at work in cancer, whether induced by treatments like chemotherapy or immunotherapy or particular vulnerabilities that are created by the dysregulated processes that give rise to the biology of cancer, allows us to target these pathways to improve the outcomes for patients. And while we're using or combining existing therapies for better effectiveness or developing new therapies designed specifically to leverage a particular mechanism, this knowledge is helping us to create better treatments. Whether we can kill more cancer cells or kill cancer cells more selectively compared to normal cells or target particularly aggressive cancer types, all of these are the outcomes that we want to pursue. One of the cell death pathways that came up most often when we think about cancer is apoptosis. Some people call it programmed cell death or cell suicide. And it's a mechanism by which chemotherapies or radiotherapy typically induces cell death using traditional cancer treatments. Now, similarly, with the advent of immunotherapies, there's been a lot of interest in T-cell mediated cell death, which is the mechanism by which those medicines work. Now, this is a process where T cells kill other cells, often specific cancer cells or specific target cells. Ferroptosis, indicated here, is an iron-dependent form of cell death and one of the most recently discovered cell death pathways. There are several potential ways that we could leverage this mechanism against cancer cells, and we're going to hear a lot about this later on from Whitney. And finally, I want to talk a little bit about immunogenic cell death, which is a form of cell death that has been postulated to trigger an immune response. My lab has been interested for the last decade in trying to understand this process and thinking about whether we might be able to harness it by combining chemotherapy with immunotherapy in the treatment regimen for patients. Now, the overall response rate of solid tumors to immunotherapy, to immune checkpoint blockade-based immunotherapies, is terrible. Immunotherapy is a terrific treatment for certain liquid tumors, certain tumors of the blood. If you have a particular type of solid tumor, a colon tumor that is what we call mismatch repair defective, immunotherapy is the cure. But for most other solid tumors, immunotherapies have just not had a very good track record. It is getting better, but it leaves a lot to be desired. Now, some of the approaches to doing this are based on conventional dogma that says that chemotherapy treatments can induce what's been called immunogenic cell death by causing these dead cells to release certain factors. We call them damage-associated molecular patterns, or DAMPs, that should stimulate an immune response. So combining checkpoint blockade with chemotherapy, you would think should make it work, right? In clinical trial data that I looked at, extending up until January of last year, I could find 15 phase 2 or phase 3 clinical trials that combined chemotherapy with immunotherapy and had an appropriate control group. And I'm sorry to tell you that out of those 15 trials, only one resulted in any benefit beyond 4 months, and that was in non-small cell lung cancer. Here's my favorite example of a beautifully done trial that gave us the exact results we did not want to see. This is a trial in ovarian cancer. My lab is very heavily focused on ovarian cancer and certain types of colon cancer. And when this trial, you're looking at patient-free survival here on the y-axis and time on the x-axis, and these are patients with ovarian cancer who were treated with standard of care. That is, they got a platinum DNA-damaging drug, and they got a taxane, an antimicrotubule drug. And they either got the drugs alone, or they got the drugs combined with immunotherapy, or they got the drugs followed by immunotherapy. In green are the patients that got the chemotherapy followed by immunotherapy. In blue are the patients who got chemotherapy with immunotherapy. In red are the patients that got chemotherapy alone. You can see that the patients that got the combination therapy did even worse than the patients that just got chemotherapy alone. We have got to find a way to be able to do better. And so in some initial studies that we did, and I'm going to summarize many, many months of work very quickly to give you an idea of our initial efforts behind understanding these disappointing results in the clinic, we developed an ex vivo method to assay whether chemotherapy, DNA damaging or antimitotic chemotherapy, would induce immunogenicity. And we found that a wide variety of chemotherapeutic drugs were able to activate naive CD8 T cells and make them immunoreactive. And in particular, in the case of a common type of chemotherapy called topoisomerase inhibitors, we were surprised to discover that the immunogenicity that this chemotherapy caused was very drug dependent, it was very dose dependent, and it did not correlate in any way with immunogenic cell death. What I'm showing you in this plot is T cell activation. So the bigger the black bar, the more immunogenic the stimulus was, using a topoisomerase inhibitor called mitoxantrone. I'd like you to notice that only these low doses seem to cause immunogenicity. These high doses you see here cause no immunogenicity. In fact, these high doses were the doses that caused cell death. The lower doses, the doses where we saw immunogenicity, gave us a mixture of live cells and dead cells. And you can imagine our surprise when we fractionated those cells and we asked, was the activity, what was being immunogenic? Was it the combination of live and dead cells? Was it the dead cells? Was it the supernatant from the culture media? No. All of the activity, all of the immunogenicity was in the live injured cells, not the dead cells. In fact, we were able to show that we could use one of the more common types of chemotherapy, doxorubicin, and a variety of other agents, all of which are capable of inducing immunogenicity in tumor cells, if you use them at the right dose. If we give up on this concept of immunogenic cell death, if we say it's not dead tumor cells that are releasing these damps that's causing activation of cytotoxic T lymphocytes, instead, it's another concept. It's the idea of immunogenic stress in live cells that is capable of inducing anti-tumor immunity. Now, you could say to me, well, that's great, Mike, except that's all cells in a dish. How about real tumors in a real model of cancer? And so we did that as well. What we did in this experiment was we took mice that had tumors, those are the black circles, and we either injected the tumors with saline or we injected them with chemotherapy to induce DNA damage, and then we added a checkpoint blockade, the very same treatment that we would give to patients, anti-PD-1 and anti-CTLA-4, and we measured the size of the tumors as a function of time. And I'm sorry to tell you that if you inject a DNA damaging drug directly into the tumor, and then you give checkpoint blockade, compared to checkpoint blockade alone, you don't really get much of a response. But hey, wait a minute. Injecting DNA damaging drugs directly into the tumor, that's like the clinical trial, where we're giving the patients chemotherapy and then we're giving them immunotherapy, we're not seeing a response. Okay? The surprise came when we took the tumor cells, we took the tumors, you see them here in black, and we took a piece of those tumors and we treated that with chemotherapy. And then we injected those live injured cells as an adjuvant back into the tumor. And then we treated those mice with immunotherapy. Remarkably, a subset of those mice developed complete regression of their tumors. In fact, 40% of those mice were completely cured of their cancer. This came as quite a bit of a surprise to us. But in those mice, in fact, we could show that they had circulating in their blood T cells that were directed against the tumor. We had been able to use this tumor as a way to convert a non-immunogenic state into an immunogenic state by using live, damaged, but not dead tumor cells. Now, remarkably, not only did this cure the mice of the tumors, but it conferred immune memory and it conferred something called epitope spreading. We could take those mice that were cured, or we could take naive mice, and then 150 days after they had been cured of their tumors, we could re-inject tumor cells into the mice, re-challenge them with live tumor cells. All of the naive mice grew tumors. That's what you see in these circles. In the squares are the mice that were cured. They were completely resistant to re-challenge with those tumor cells. Furthermore, we could take the spleens out of those mice, and we could examine them for whether or not they had developed additional T cells against the tumor, and that's what I'm showing you here. This is everywhere you see a red dot, that's a stimulation assay, and you're looking at activated T cells. And in fact, not only could we use the tumor cells that we had induced the immunogenicity in, but we could show that they had developed an immune response against the parental cell line itself. Not only had we been able to induce immunogenicity against an antigen that was on our tumor cells, but they had developed T cells that reacted to additional antigens that were present. Okay. So what this told us was that now we have a mouse model and we have a new idea of an immunogenic cell pathway that doesn't actually work like people thought it did. So I think these results are pretty interesting. What it really says is that in a tumor to begin with, you have a bunch of exhausted T cells, those are the green circles, and a bunch of stressed out antigen-presenting cells, those are the blue star-shaped things, and you have a bunch of live tumor cells. If we continue to do exactly what the clinical trials are doing, which is the equivalent of injecting chemotherapy into a tumor, yes, we will get some live injured tumor cells and a bunch of dead tumor cells, but we're damaging the T cells and we're damaging the antigen-presenting cells. This approach is why the clinical trials aren't working. What this is telling us is that, in fact, if we use a different approach, we find a way to be able to induce injury but not death within the tumor itself without damaging the microenvironment of the tumor, then using something like this injured cell adjuvant, we can enhance the function of antigen-presenting cells in the tumor, and we can enhance the response of T cells against the tumor.
All right, good evening, everyone. I hope those in the back can hear me clearly. So I want to begin by highlighting that while we have made significant progress in improving cancer survival over the last several decades, the sad reality is that in 2025 in the US, there will be an estimated over 2 million new cases, and over 600,000 individuals will still die from this disease. That translates to about 4 new cases and 1 cancer-related death every minute. One of the long-standing challenges that accounts for this high cancer mortality is the failure to effectively eradicate subpopulations of cancer cells that are highly metastatic and notoriously hard to kill using our current anti-cancer therapies, ranging from your classic cytotoxic chemotherapies to targeted therapies like tyrosine kinase inhibitors and even more advanced immunotherapies. Therefore, the ability to target these subpopulations of cancer cells will be transformative, and it will bring us one step closer to achieving more durable responses in the clinic. Today, I would like to share with you one promising strategy that really exploits the heightened susceptibility of these therapy-resistant cancer cells to a relatively newly described form of cell death called ferroptosis. So the word ferroptosis was coined in 2012. And as the name implies, ferroptosis is an iron-dependent form of cell death that's largely driven by a buildup of oxidized membrane phospholipids. This increase in iron-driven oxidation of membrane phospholipids leads to plasma membrane thinning and destabilization, significant cytoskeletal remodeling, and this distinct ballooning-like morphology. I guess my video doesn't work well. This distinct morphology that has this ballooning-like shape that's distinct from the classic blebbing observed with the more well-studied form of cell death called apoptosis. So what you should have been seeing in this live cell imaging video, which somehow doesn't work right now, is cancer cells undergoing ferroptosis. Now, to prevent spontaneous induction of ferroptosis, many cell types rely on key lipid antioxidant pathways. GPX4, glutathione lipid peroxidase 4, is a key enzyme that protects most cells from undergoing ferroptosis. And it does so by recruiting an antioxidant called glutathione to help the cell reduce lipid peroxides, which are highly reactive, into lipid alcohols. Now, inhibition of GPX4 using, say, small molecule inhibitors leads to this buildup in these highly reactive lipid peroxides. And in an iron-dependent manner, toxic levels of lipid-reactive oxygen species, which if left uncontrolled, ultimately causes the death of the cell via ferroptosis. Here I want to highlight that iron plays a critical role both in the initiation and the propagation of this lipid Ross cascade, and that polyunsaturated phospholipids, which are an important part of our cell membranes, are considered the key substrates for lipid peroxidation in ferroptosis. Given the importance of iron and lipid Ross in driving this mode of cell death, ferroptosis, in this example, in this cell viability assay, induced by treating cells with a GPX4 inhibitor, can be prevented by co-treatment with an iron chelator or a lipophilic antioxidant, but not a pan-caspase inhibitor or a pan-RIP kinase inhibitor, which would be indicative of other modes of cell death. Seminal work out of the lab of Stuart Schreiber over at the Broad have highlighted that cancer cells that have what we call a high mesenchymal gene signature show a heightened susceptibility to ferroptosis. And this includes cancer cells like sarcomas that naturally have a mesenchymal gene signature. And these sarcomas represent a significant clinical unmet need. We have also, consistent with their findings, observed that epithelial breast cancer cells that have now acquired a mesenchymal phenotype are also highly susceptible to ferroptosis. And we are particularly interested in these mesenchymal breast cancer cells because they tend to be highly metastatic and they are notoriously hard to kill with your classic cytotoxic chemotherapy and more recently shown to be highly immune evasive. So these tumors tend to have poor response to immune checkpoint blockade therapy. We are also exploring the potential of ferroptosis in Merkel cell carcinoma and in Ewing sarcoma with some of our colleagues over at Dana-Farber. Now, in the ferroptosis field, we have really good drugs to induce ferroptosis in vitro, meaning in cells. But there is this urgent need to identify novel ferroptosis-inducing compounds with good pharmacokinetic properties and high potency that can work well in vivo, in animal models, because these are going to be very important to really determine if this strategy that I'm telling you really works. And so we have decided to do something about that. And so we have established a collaboration with the IOCB in Prague, and this is being spearheaded by one of the graduate students in my lab, Ryan Mansell. We have just completed the first phase of our ferroptosis-inducing drug screen, and this screen contained a compound library of over 12,000 compounds. This involves compounds where we have novel chemical scaffolds, natural compounds, steroids, peptides, and so on. And we are happy to report that we have some very interesting leads. We don't know their structure yet or how they work, but that's a good start. In the meantime, we decided to use a genetic approach to ask what happens if you knock out GPX4 in mesenchymal breast cancer cells in vivo, and what effect does it have on tumor growth kinetics? And here is where we found some interesting and unexpected findings. We observed that implantation of ferroptosis-prone GPX4 knockout cells into the mammary fat pad of syngeneic immunocompetent black 6 mice led to a greater reduction in primary tumor growth kinetics and tumor weight compared to when we implanted these cells in immunodeficient NSG mice, which lack T cells and B cells. This was the first hint that loss of GPX4 might be eliciting an adaptive immune response, or at least eliciting adaptive immune cells to mediate its anti-tumorigenic effect. When we took a closer look at these tumors, we observed that there is very little staining for cleaved caspase 3, which would suggest that there's very little evidence that apoptosis is occurring. However, we do see indications of cell death via tunnel staining, likely ferroptosis, but we still need more, I would say, to really confirm that. We also see increased staining for lipid peroxidation byproducts in our GPX4 knockout tumors. Another piece of evidence that potentially lipid ROS is increasing and maybe ferroptosis might be occurring in these cells. And we have more to do on that end. And this immunohistochemistry analysis was spearheaded by a talented research assistant in my lab, Navia Kapadia. Now, all of this is still very correlative. How do we functionally test that CD8s and CD4 T cells are important for our phenotype? And so what we learned through a series of depletion assays is that if we deplete CD4s and CD8 T cells in the context of GPX4 knockout implanted cells, this actually leads to an outgrowth of GPX4 knockout mesenchymal breast carcinomas. So another piece of evidence that these CD8s and CD4 T cells are functionally important. We then went on to take a closer look at what's happening inside of the tumors that might give us further confirmation that these cells are important. Oh, and we saw very little effect with macrophages, or the opposite. So here what you're looking at with this immunofluorescence is the tumor border of wild-type mesenchymal tumors versus GPX4 knockout mesenchymal breast carcinomas. And already you can appreciate this increase in T cells in the GPX4 knockout tumors. These are the CD3 positive magenta colors that you're seeing on your screen. Tumors are in green. If we zoom in and we go now into the interior of the tumors, again, you could see that these wild-type mesenchymal breast carcinomas are largely devoid of T cells, whereas, again, we see this increased infiltration of T cells in the GPX4 knockout tumors. And this is all through CD3 positive staining. We then wanted to take a closer look and really look at the type of T cells. And here we performed IHC. And again, you could see that there's an increased infiltration of CD4 and CD8 T cells in our GPX4 knockout tumors. Now, when I started off, I said to you that mesenchymal breast carcinomas are highly resistant to immunotherapy. And that is indeed the case. If you compare the black dots and the light gray, they're pretty much the same. These tumors show very little effect or response to treatment with anti-CTLA-4 immune checkpoint blockade therapy. But because we had seen this increased infiltration of T cells in the prior data and that the tumors were smaller, we wondered if we could capitalize on that and sensitize these cells to immune checkpoint blockade. And here we decided intentionally to do longitudinal studies. This revealed that loss of GPX4 in combination with anti-CTLA-4 leads to durable responses. And we followed these mice for over a year. I also don't want to ignore the red dots. And that is because we did these studies for so long. After about 50 days or so, you do eventually see subpopulations of GPX4 knockout cells that do eventually grow out. And so our lab wants to know why. And how do we prevent that from happening? We also took it a step further, and then we performed experimental metastasis. What that means is we implant the cancer cells via the intracardiac route and allow the cells to disseminate and colonize wherever that cell has a propensity to grow out. And here, again, we see that knockout of GPX4, either alone or in combination with anti-CTLA-4, leads to a significant reduction in metastatic burden. What you are looking at is histological images of kidneys from mice implanted with wild-type and GPX4 knockout cells. Now, while all of this is intriguing and it in many ways supports the promise of ferroptosis-inducing drugs, we believe that in order to realize the full therapeutic potential of ferroptosis, it is critical that we first acquire a fundamental understanding of the biochemical molecular pathways that regulate ferroptosis and its biological roles within physiological context. And so part of my lab really is addressing some of these very fundamental, basic biology questions. Already we have begun to get some new insights into what are some of the types of organelles that are important for ferroptosis and in what context are they important. And so we've had some insights into the importance of the peroxisomes in modulating ferroptosis susceptibility. We've learned about certain types of understudied lipids and how they can affect the pool of substrates that undergoes lipid peroxidation, as well as the availability of redox active ion via changes in membrane biophysical properties. And more recently, we've also gotten some insight into some new molecular scaffolding machinery and enzymes that coordinate the lipid remodeling associated with this heightened ferroptosis susceptible state. We are also invested in looking at what are the effects of ferroptosis in vivo. What effect will it have on the tumor microenvironment? And so, more specifically, we are interested in asking, does the tumor microenvironment affect the susceptibility of the cell to ferroptosis? What happens if we implant these cells in the brain versus the liver versus the lung, where the metabolic composition is different, the cellular composition is different? Is that something that we should take into account? We also are interested in the impact of ferroptotic cells on the tissue milieu. What factors are secreted when these cells undergo this mode of cell death? And we like to think not just about the cytokines. We want to think about oxidized lipids, metal-binding proteins, metabolites, and so on. And what is the impact of these secreted factors on the activity, the viability, the recruitment of various immune cells and stromal cells, and how that might affect tissue architecture and the function of that organ. And most importantly, how do we begin to leverage these findings to overcome ferroptosis resistance in vivo, using both hypothesis-driven approaches as well as unbiased functional genomic screens? And so we're now building our lab and our team, and we're excited to see where this goes and what new insights we can have to help to inform how we can get rid of these really hard-to-kill therapy-resistant cancer cells. And so this brings me to what I think is the most important slide in my presentation, the acknowledgements, all the people who did some of the heavy lifting. I'd like to thank the members of my lab, and you could see some of them right here. We've now started to increase in numbers. All the collaborators, both internationally and nationally, and the fantastic core facilities here at MIT, and our sources of funding from the Bridge Project and the Emerald Foundation. And I thank you for your attention.
Okay, so at this point, I'd like to invite all three of our speakers up to the front, and we're going to open it to questions. This is my script, but there was a script up there that had time, questions, all that. It's okay. Questions? Please.
In the context of the epithelial ovarian cancer, I wonder, when those ovarian cancer, they have BRCA1, BRCA2 mutation, and they already had a, impaired, mechanism, and why they don't undergo, that kind of suggestion, at a stimulation on the environment, had that kind of stress or damage. Before we need to do the apply drug, PAP7 inhibitors.
So it's an excellent question, which is, after all, Many of these cells, particularly in ovarian cancer, are defective in proteins that are involved in DNA repair, and so they probably already have some DNA repair. How come immunotherapy doesn't work? So one person whose name was on the acknowledgments who I didn't acknowledge was actually Tiffany's PhD advisor, Bram Siegel, who was at Roswell Park and is now at Moffitt Cancer Center. And together with him, we've been studying the tumor microenvironment. And in fact, one of Tiffany's earlier work shows that within the microenvironment of ovarian cancer, neutrophils, inflammatory cells have become activated, and those neutrophils are directly attacking T cells, and they're converting those T cells into a state of functional paralysis. And so I think that we are able to overcome that in some way by this ex vivo DNA damage in live stress cells. But it may very well be possible to target the signals that are activating those neutrophils in the microenvironment and make immunotherapy work better. And in fact, we have a clinical trial in progress with Roswell Park testing just that exact idea.
Oh, wonderful. Thank you.
Please.
Thank you so much. I'm curious, you hear all about AI in drug research. Do you use AI in your work?
Let's see, is it still on? Great. I think you're thinking exactly where we would like to head to, especially with the drug designs. So a lot of our collaborators and the medicinal chemists who are going to follow up on many of the leads that we found from our screen will be using a lot of that technology to be able to hopefully improve pharmacokinetic properties, tumor targeting specificity, and things of that nature. And I think that's exactly where we should be heading.
So we've always had an interest in using computational methods combined with the experimental methods to try and get at what's going on. So at the moment, we're in the process of building a lab-focused large language model that we're in the process of loading up with what's out there in the literature related to DNA damage and immunology and feeding our data into it. We don't have results yet, but that's exactly what's ongoing right now. An excellent, excellent question.
Hey, so I had one question about the live but stressed tumor cells. I know from working with dendritic cells, you can irradiate them so they're functional and alive but can't replicate. Does it work, or have you tried doing that with the live but damaged tumor cells?
So we get this question all the time. We have not tried to irradiate the tumor cells yet. I fully believe it'll be very similar. As long as you're using a dose of radiation that you're either using small doses over time or something that just doesn't completely kill the tumor cell. But I think it will be very similar mechanisms.
What you're getting at is what's called, as you probably know, the abscopal effect, and that's what's seen in certain patients whose tumors get radiation. So if someone has metastatic disease and you radiate, for example, a tumor that's destroying, let's say it's destroying the humerus in the arm, sometimes you find not only does the radiation induce death in the tumor that's metastatic, but you begin to see tumors elsewhere in the body begin to regress that were never irradiated. And that has always puzzled radiation oncologists. It's called the abscopal effect. And I suspect it's exactly at what you're getting at. There's some component probably of live injured cells that are able to activate the immune system. We actually have a collaboration that we're hopefully make more progress in looking at exactly that question.
And then I had one other question. I don't know a lot about all the different types of cells out there, but I know epithelial cells can sometimes have niches where they are presenting antigens. And so I'm wondering if you were to use the same approach against, I don't know, maybe muscle cells don't do as good of a job. Do you think this would still work?
Yeah, I think you have to find the right environment for the cell, the right amount of damage, and whether they have increased expression of signal 1 and signal 2 that I talked about. If a cell can do that, I think any cell could perform this function, but we've also seen there's a very narrow window that they can directly activate T cells.
What's interesting is if we take those damaged tumor cells, so I showed you that if we took those damaged, live, stressed, but damaged tumor cells and injected them back into the primary tumor, that generated a pretty good immune response. Sriram also did an experiment where he took a mouse that had a tumor, took those live, injured, damaged tumor cells ex vivo, and injected them into the opposite flank, into a lymph node in the opposite flank, instead of into the tumor itself. And then the immune response to the tumor was decreased. It was still there, but it wasn't as potent as it was when the tumor cells were injected into the tumor, which makes me think that there's an injury response from the very act of injecting something into the tumor. You know, immunotherapy started with a crazy, an insane observation by a surgeon, a guy named Coley, who was taking heat-killed bacteria, and he was injecting them directly into the tumors. And some of his patients were cured. In fact, Merck, for decades, made this stuff called Coley's toxin, which was a heat-killed bacterial toxin that people would inject into tumors. And I do think that there's something that happens from the very act of injection that itself may be modulating the immune system, and that we have not looked at directly, but I have a feeling that that may also be involved in, for example, why surgery sometimes appears to make dormant tumors become more active. I think we really don't understand enough about the injury response, and I think it gets at a lot of important questions.
Thank you.
Please.
Hi, first of all, thank you so much for the great talk. So I do a oncolytic virus research at Brigham Young Surgery. Do you think that oncolytic virus, the question is kind of two parts. First part is, do you think oncolytic virus kind of induces a similar state into this DNA damaged cells? And the second part is, if it is this intratumoral injection of damaged cells is useful in brain tumors, what do you see as the translational barriers to it?
Two excellent questions. Oncolytic viruses, your thoughts?
I haven't even thought that hard about it. Well, right now we're still trying to figure out what are the physiological triggers of ferroptosis, a big outstanding question in our field. So potentially that might be one of those species where we could do some further investigation. I would say potentially, if it's affecting many of the key sort of lipid antioxidant pathways, if somehow that leads to a down regulation, then we're exposing a vulnerability, or if that is in some way affecting the lipidome, where your cell now somehow becomes more polyunsaturated for whatever reason, then again, I could see a link here. But when I survey the landscape of the types of contexts in which we study ferroptosis, it seems like it's still a little bit more limited. But I think there's growing interest in our field, so maybe it's just a matter of time before we can pinpoint the mechanism here.
Now, you asked about translational barriers. I think about this a lot. Can you imagine if I went to an institutional review board and I said, here's what I want to do. I want to cut a piece of this patient's tumor out, and then I'm going to treat it ex vivo, and then I'm going to inject their own tumor with parts of their tumor that I took out and manipulated. I don't think a single IRB is going to have an easy time letting me do that. Now, it doesn't mean it's not the right thing to do, and I think it's worth pursuing, especially because we can say it's the patient's own tumor, right? We're not, right? Especially if, as Tiffany continues to point out to me, we can prove that the manipulated tumor cells that we use can't proliferate any longer. But I think another real way to get at this is antibody-drug conjugates. We know that there are a number of antibody-drug conjugates out there. In fact, some have been approved, one in particular for ovarian cancer. But the two payloads for antibody-drug conjugates, the idea behind antibody-drug conjugates is you try to make your chemotherapy only work or preferentially work in the tumor cell by conjugating it to an antibody that recognizes something on the tumor cell. So the tumor cell then engulfs the antibody, takes in the drug, and preferentially gets killed, in contrast to any other cell in the tumor microenvironment. The two classes of antibody-drug conjugates that are out there for cancer, One are antimicrotubule drugs linked to an antibody. But the other are topoisomerase inhibitors. The exact drugs that I've shown you are capable of inducing immunogenic cell death if used properly. And so my hope is that perhaps we'll be able to start a trial asking whether if we use antibody drug conjugates, topo-1 conjugated antibody drug conjugates, whether those with immunotherapy might work better than the systemic immunotherapy that I've shown you does not seem to work.
Thank you.
Please.
Thank you for the very nice presentation. So I have two questions. First for you, Dr. Yaffe. When you injected those damaged tumor cells back into the tumor and you improved their response, you still had non-responders.
Yeah.
So have you made any attempts at characterizing and profiling those responders versus non-responders to understand the difference? How do we make all of them respond?
That's a very astute question. Yes, 60% of those animals did not have their tumor disappear. What's different? These are inbred mice. How can they show such a diversity of responses? That's a terrific question. We have not begun to work on exactly what that is, because what we're trying to do, but it's something we have to address. I think what we're trying to understand from work that Tiffany's doing is what exactly are the signals so that we know how to combine. Right now, it's guesswork. It's okay, we tried a bunch of drugs, we saw what worked in vitro, we used those drug doses with the in vivo aspect of this. If we could have a marker that said, this is the pathway that's being activated, then we could find a whole bunch of different drug doses that would work, right? Or a bunch of different drugs that might work at different doses. And then we could begin to ask in a unified way, what's wrong with the other percentage of mice in which the same treatment doesn't work? But that's a critical question that you point out, absolutely. Your second question.
So my second question is for Dr. Henry. So you've knocked out GPX4 constitutively and inoculated it either sub-Q or intracardiac. Have you tried to generate an inducible knockout and actually knock out GPX4 once you've established a tumor to see if you can then impact metastatic dissemination?
Yeah. Well, it seems like you're overseeing, you've been a fly on the wall in our lab. That's exactly, I think every student in my lab is working on some strategy to get an inducible system going. In the meantime, when we implant these GPX4 knockout cells, we implant them with a lipophilic antioxidant. And the idea is that what we're implanting is viable cells, and then over time, because we're not replenishing these mice with lipostatin or any ferroptosis inhibitor, eventually we will unmask the dependency. But that's exactly where we're going here. Yeah.
Please.
Along the same direction, just follow up the previous question. And, very nice, you point out the key factor of the T infiltration, tumor infiltration lymphocyte is a key pair. I did one thing. Instead of, inject the damaged T cell or some other, white cell, I took part of the tumor cell, do a biopsy, microbiopsy, take part of the tumor from the patient. Yeah. I'm not doing rodent or chasing monkey. I just go directly to the patient. Yeah. And then we send that, a tiny piece of things to a company outside and let them, do the teasing apart, and we try to isolate those, small amount of TIL. Yeah. And then after the, a process, we purify and put it back to the patient, and at the same time treat it with a low dose of interleukin. I forgot whether it's 1 or 2, but it activates. The patient don't like it because make it feel flu-like syndrome. Almost they feel I get the flu, am I getting the flu? But but that kind of treatment, activated TIL, and this is a very old model from NIH. I was
from
there
Rosenberg.
from Rosenberg. You know that more than I do, but it worked.
Yes.
But same thing is true. You have responder, partial responder. Well, something might not that desirable, but you still have something on the positive side. So it's still a mystery, and we need to compare different way of doing things. But the key prayer is still the TIL, tumor infiltrating lymphocyte.
No, that's a very good point. So what he's referring to is the early work not early work, continued work from the Rosenberg lab, that if you harvested tumor-infiltrating lymphocytes, TIL, and you could culture those and then re-stimulate those and reinfuse those into the patient, perhaps you would get a particularly good response. And it is true, you do see some response. There's a lot of toxicity because you have to typically give the patients a pretty hefty dose of interleukin at the same time that you infuse their T cells. But, and again, as you point out, it only works in certain patients for reasons that are still relatively obscure. Now, I can tell you two things that, because Steve's a friend, I can tell you two things that are happening. One is they're beginning to use this using CAR T cells, using chimeric antigen receptor T cells, by engineering those against tumor antigens and then trying to see if they can use some combination of CAR T cells and TIL to increase the ability of this type of immunotherapy. But it does also suggest, though, the possibility that perhaps if we could combine, the beauty of tumor-infiltrating lymphocytes, right, is they at least got to the site. Why they don't work, we don't know, but they're at least there. Perhaps some combination of the type of live but injured cell stimulation plus stimulating tumor-infiltrating lymphocytes might work. The other thing, of course, that we've learned from this, again, I see this done the right way and the wrong way, right? We think, okay, let's harvest, let's do surgery, resect the tumor, and then we'll give immunotherapy, right? And that'll kill any of the remaining tumor cells. In fact, that's the exact wrong way to do it. Why? Because when you harvest the tumor, you're taking away all of the T cells that have managed to home to the tumor in the first place. What we really ought to do is give immunotherapy and then resect the tumor and then finish with more immunotherapy. That is beginning to be done at some places, but the vast majority of places, I think, are still doing it backwards, which is the surgery first, then the immunotherapy. No, that's a mistake. One more question.
Mike, thanks. Wonderful talk.
Thank you, Yogesh.
So now I'm thinking about the product market fit, because there are several types of cell death and there are several types of tumor types. Is there any data which can correlate, like, oh, this particular type of cell death is fit for this particular type of tumor, or it can be preferable match between the type of cell death and type of the tumor type?
You mean for immunotherapy? You mean
yes.
I'm going to turn that over to you.
So it's basically, the question is, are there specific targets after a specific cell death?
Are there certain, should you say, oh, this is cervical cancer, we should use this drug to cause immunogenic cell death, or this is thyroid cancer, we should use this drug? Thoughts on that?
So far, we've seen using the same drugs in a murine melanoma, a murine colon carcinoma, and a murine ovarian cancer cell line. It seems to be a generalized effect where as long as you have enough stress going on inside the cell, your body can recognize it. So I'm not sure, there probably might be some cancers that don't respond the same way, but at least for ones that we've tried, I think it's more of a general injury response or stress response that's leading to your immune cells recognizing these tumor cells and getting rid of them.
I think because I have a sort of an unusual way of thinking about this. I think because I trained as a trauma surgeon, right? And I think the thing that we forget, we're all taught that the immune system is designed to recognize non-self and be tolerant to self. But I believe much more in a different immune model that Polly Matzinger put out at NIH many years ago called the danger model. And what Polly's model says was it's not self and non-self. It's It's danger versus non-danger. And after all, our immune system, the thing that it confronts all the time is injury and stress. And our immune system is probably programmed not so much to respond to dead cells. Why would you care about dead cells? They're already dead. But to respond instead to those really stressed cells that you don't want to persist. And perhaps what we've stumbled onto here inadvertently in trying to combine chemotherapy with immunotherapy is more of what the function of the immune system is under conditions of normal stress, which is to recognize those stressed cells, not the dead cells, and take those out.
My one last question from my side is, Mike, thanks to AI, that there are lots of new targets, new molecules, and as an investor, we're telling that please don't bring any new target or new molecule because it's very kind of time-taking and capital-consuming to prove those target and those molecule as a product. So what's your thought around how we can repurposing, like, you know, how we can repurpose those drug which is already in the market and induce those cell therapy, you know, the cell death, and use that as a new modality?
So I'm going to turn part of that question over to Whitney and ask her where we are with drugs in vivo that are going to activate ferroptosis.
Good question. So we only have about 3 that I can think of, and they indirectly inhibit GPX4, so they're not site activated. They don't really attack the catalytic side of GPX4. So we still have a long way to go, I would say, in terms of the ferroptosis field. I'm hoping that as we do more drug screens and so forth, potentially because our screens do have natural compounds and some of it is FDA-approved drugs, there might be drugs that we had not considered that might induce ferroptosis and we can repurpose, but this is still a wide open area for further exploration. I think for now what our field is doing is getting the proof of concept experiments. Is this really a promising strategy? How best to use it? What's the context in which you should use it? What's the regime that you should use it in? As we're learning with Mike's data, timing is important. The context is important. And so where my lab falls into that is to really do all of the dig deep in and getting us the truth around the fundamentals of ferroptosis research, so that when we get the drugs, we know how to best use them. When I think about what we learned from the tyrosine kinase inhibitors and the early trials, we knew that they could do their job, but some of the earlier trials failed because we didn't know that we had to classify the patients in a given way. We thought monotherapy would have been one way and then learn combination therapy. Then we had to learn how do we do the scheduling, and all of these things mattered, and they determined whether some clinical trials worked and some failed. And so I think even people like us who come more from the basic biology end do play a very important role in really determining the outcome of such potentially new therapeutic approaches.
Okay, and I'm going to thank my colleagues and thank you for your attention and engagement.